Hi Year 11, so we're just going to have a quick look at this question together. It's a paper 1 question from 2020 and it's the 6-mark question asking about DNA. So it says DNA is found in the nucleus of cells. Describe the structure of DNA and how it can be extracted from plant cells. So it's sort of split into 2 separate parts here. So describe is to it will tell the examiner about the structure of DNA, so we'll have a think about what we're going to be including there, and then how you can extract DNA from plant cells. So in terms of knowledge, first of all, just notes, hopefully remembering that DNA has a double helix structure, meaning it's made of 2 strands. It's made up of 4 bases. So we've got adenine, thymine, I'll write them as the pair up, so A with T, cytosine and guanine. And it's okay just to refer to the letters. So we have complementary base pairings here, so between adenine and thymine, which basically means you will get either that pairing or that pairing, and cytosine and guanine are complementary base pairs, so C with G or G with C, and they are called complementary, meaning to complement, so complementary base pairings, and they are held together by weak hydrogen bonds. You could also mention that DNA is a natural polymer. So that's something else that you could put in your exam question, but we've mentioned structure, base pairings, you could add nucleotides, and you could talk about the phosphate sugar backbone as well. So in terms of what we would want to write down for the extraction, so I'm running out of room here, so I'm going to have to put it on the side. So for the extraction, we would talk about to crush or to grind the plant cells, to break down any cell walls, and then you'd want to talk about adding soap or detergent and salt. So the soap will help break down the cell membranes and the salt will make it stick together. And then you would filter the solution, and any insoluble parts will stay inside the filter paper and the filtrate will pass through. And then you would want to add ice cold or pour ice cold ethanol down the side of the solution, and that will cause the DNA to precipitate. And the word precipitate means when a solid forms within a solution, and it means that the DNA is now visible. So how would I write this in an exam question? So I would bullet point it, and I would say DNA is a double helix polymer. It's made of 4 base pairs, and then I would write them down, A with T and C with G, and they are held together by weak hydrogen bonds. So we've said double helix polymer, we've mentioned our 4 base pairs held together by weak hydrogen bonds, and then for the extraction part, you want to write down that you would crush or grind up the plant cells. So crush, grind up the plant cells, and you'd use a pestle and mortar to do that, and then you would add detergent or soap and salt, and that will break down the cell membranes, including the membrane around the nucleus, and salt will make the DNA stick together as well. And then you would filter, so you would filter your solution, and what you want to do is you want to collect any of the insoluble parts inside the filter paper, and then the filtrate will then pass through. So filter and heat in a water bath. And then finally you would pour ice cold ethanol down the side of the boiling tube so the DNA can precipitate. And if you can't remember that word, just say become visible, which means you can see it. So just in terms of the mark scheme, what the examiners are looking for, so I'll just show you here for structure of DNA. So we've talked about polymer bases, how they would pair up, weak hydrogen bonds, 2 strands, and then you've got your DNA extraction on here. And then some additional guidance. So you can see here, so we're sort of aiming for 5 and 6 on these questions here. So have a bit of a read through and just see what you could add to your answer. Okay, hope that helps. Well done.